Hi, everybody. This is Mr. Crook again for Botany and Zoe. So, what we're going to do is look over project number 5, uh, so that way you guys can get a little bit of help with this. There is a couple questions here that students have gotten wrong in the past, and I just want to make sure we are scientifically accurate as best as we can be with this. Um, this is an interesting one because with these two parts to project number 5, project or part 1 from lesson 22 basically goes over the respiratory system, looking at the lungs and how lungs work. And then from lesson 24, you're looking at the 3-chambered hearts of amphibians and lizards. So we're going to go over this a little bit because it's this question on lesson 24 that people are getting wrong. Or they just don't understand, and when I'm reading it, I can tell that they just don't fully get it. And so I want to go over this very quickly, and I'm actually going to go into a little bit how the textbook or this lesson is kind of wrong, and how it kind of explains the 3-chambered heart, and that question is misleading. Okay, so I'll go over this, and you guys can get full points on this because of this walkthrough. So here we go. Okay, so when we're looking at this, this is for the first part. I'm just going to kind of briefly go over a back view of lungs and gills and how they kind of work. I think you guys got this down, but we'll go over it just in case. So when we're looking at this, here is a fish, obviously, and you can see kind of the breakdown of how these gills work. Gills are very fascinating, and they have adapted very well to the environment that they're in. And they are very efficient in getting that oxygen out of the water and getting it into their blood. And some things that really fascinate me, if you look how many gills they have. Okay, so as it goes through, and you can see how kind of the water moves in and out and weaves through there to get every last bit of oxygen that they can get. And this occurs via diffusion. Okay, and so it is really cool, and it's using natural laws to do this. And so what we have here is these are the gills broken down, kind of shows you how the water kind of sifts through that. And as it sifts through that, you can see the gills that are look how much surface area there is. Surface area is key when we're looking at this. The more surface area there is, the more oxygen they're going to be able to diffuse through. The other thing that they've been able to adapt to and make even better is their blood. Okay, and so how they have this blood helps it to use diffusion to pull more oxygen out of the water. We know that molecules will move from an area of high concentration to low without using energy. That's diffusion. And so when we're looking at this, as the water flows across these gills, you can see that the blood it comes to first is oxygen-rich blood. And you can think, okay, why? Why would it do that? Okay, so there's obviously a high amount of oxygen in this blood, and the water doesn't. You can kind of see that the water has about 10% oxygen. I wish I could have gotten a better picture here. But as it goes through this, it's going to reach that oxygen poor blood where it's going to have a high concentration right here to a low concentration, and it's going to pump that oxygen into that blood a lot quicker. Okay, so as it goes through, it's going from an oxygen rich to an oxygen poor blood, and what that is causing is it's causing that blood to become oxygenated as it moves this direction. I hope that kind of makes sense. And sometimes I wish we were just more in person and I could really model this for you. But you can see as the water blood moves through that, at the very end, it reaches that oxygen poor blood, so that when it comes through, that blood becomes oxygenated, oxygenated to the point where it's up to 90% oxygenated at that point when it first meets the water. Okay, so that's kind of how fish gills work. Here's another picture. I kind of like this one because it kind of shows us this right here, there's a question there that'll ask you about these gill arches. These are cartilaginous parts of the lung. It's very strong, and it's just there to give support to those gills. Okay, protection probably a little bit too. So now when we look at the human lung, because there's going to be a question, you'll have you compare those, right? But here's human lung, mammals and terrestrial lung are very similar. Okay, and these are fascinating too. What this picture does not show is down here would be the diaphragm. As we breathe in, that diaphragm goes out, or it contracts, okay, which would cause it to go like this, I guess, if you can see my cursor, making there be an intake in air using volume and pressure. So it's creating more volume in here in this area to the point where, well, we need to bring air in to fill in that cavity, fill in that volume. And then when we breathe out, the diaphragm relaxes and we lose volume, and so it has to go, it pushes that air out. And that's why it's when you do the Heimlich, you would always put your fist down below the diaphragm to push up, causing there to be a decrease in volume, dislodging whatever it is in the trachea. Okay, and so that's kind of how that works there. But where this lesson's going to is inside of the lungs. So inside these lungs, what there are is at the ends of these little branches right here are what are called alveoli. And these alveoli are locations where there are capillaries, blood vessels, very thin, where when you breathe in that oxygen, again, it's using the same kind of procedure here, where it's going to move the oxygen from the air into the blood using diffusion. And so it's going to do that inside of our lungs, and it's at those alveoli where that are occurring. And it's the alveoli, it's a very, very thin layer of skin or some type of I don't know if it's skin, but it's a membrane, a very thin membrane that's covering those, allowing for gas exchange very quickly. One thing that I think I grew up in Wyoming. It's very cold there, and I remember when it would be very cold, I'd be standing outside and I'd breathe in that cold air, and it hurt. And that is one reason there. That cold air will actually cause the blood vessels to burst because it's so cold at those alveoli, because it's so close that those capillaries can break, and it's kind of painful. It's not threatening by any means, but it does hurt. But anyways, it's at those ends of those branches where that's occurring, and then you can see that the deoxygenated blood at the capillaries or at the alveoli. I need to find a picture. I can't believe I didn't put one in there. Let's look at a picture of an alveoli here. You can kind of see that. Okay, so like right here, um, you can see that. So you have those branches, and then you have these alveoli sac things, and you can see that is where that oxygen is being transferred from the deoxygenated blood to create oxygen-rich blood. Hopefully you don't have any questions there. If you do, please send me an email. It's very fascinating. If you're even more interested in it, take an Ant and Fizz next quarter from me as well. And we go into this in great detail. Okay, so on to this last part from lesson 24. This is the confusing part. I don't know if it's confusing, but it's kind of tricky, and I've always had a hard time understanding this. But when you look at the anatomy of the heart, we can see this is a very primitive heart. It's amphibians, and amphibians are kind of old in the evolution of living things. And what they had was a three-chambered heart. So they had two atria, okay, and then one ventricle. And you can see this kind of models how that blood flows in. So you have deoxygenated blood from the body, comes in here, gets pumped up to the lungs to get oxygenated, where it comes in right here into the atria, and then right here and out to the body. Ventricles have a lot of muscle on them, and so there's a lot of thrust to push it out to where it needs to go. Atria are not that strong. They kind of just let down into the ventricle where it then pumps. Okay. And so the question that it asks is, well, how the heck does a 3-chambered heart not allow the blood to mix? That's kind of in theory what it asks. And to me, I'm like, well, that's kind of misleading because it does mix. There's no doubt about it. It can't keep it from mixing. It does mix in the ventricle. They share the same ventricle and it will mix. However, you can see that because it is partitioned side to side, it pumps very quickly, that it doesn't allow it to completely mix before it goes. However, you are not going to have the oxygenated blood that gets pumped out of here is not as oxygen rich as it is in our blood, in our hearts, where we have a 4-chambered heart, where it is completely separated right here. Okay, and here's a picture that I kind of want to show you. So the lizards also have somewhat of a 3-chambered heart. However, you can see that in evolution it said, this isn't going to fly for larger animals. Frogs are fairly small, and so you get that oxygenated blood to the body isn't too hard, and it's small enough that even though the oxygen isn't as rich as it could be, it still functions. However, when we get to larger animals, lizards are still kind of small, but as you get bigger and bigger, you can see that they started to make this septum right here, this separation to start to make that 4-chambered heart to separate the oxygenated blood and the deoxygenated blood. And so, kind of fascinating how evolution has caused that to occur, because it is better. And so now we can have larger animals, such as an elephant, where you can have that oxygen-rich blood being pumped out to the body. And the more oxygen there is, the bigger the animal can be, because it gets that it has a greater chance to get that oxygen deeper into the tissue, into those cells. Okay, because remember, there's always that diffusion going on, and so high to low concentration. And so as it goes further and further throughout the body, that oxygen becomes more deoxygenated because it's releasing that oxygen out of the blood because of diffusion. And then it becomes deoxygenated and it pumps it back to the heart, where then it pumps to the lungs or gills and back into the heart again to be pumped back out of the body. Fascinating thing. Hearts are very, very cool. And so that's kind of how this works. So what I wanted to get at is there is mixing going on here. So when you answer that question, explain how in a 3-chambered heart the oxygenated blood is kept separate from the deoxygenated blood. It isn't. Okay, it's not kept separate. However, I mean, it is a little bit, but not much. To verify and to show that I am not teaching false things, here is a reliable source showing, guess what? It is going to mix. Okay, and you can see in this picture right here. Let me zoom in on it. You can kind of see this. So here we have 4-chambered heart, and you can see how great it is. How we have deoxygenated, oxygenated blood, and as it goes through, it will be very rich in oxygen, but as it goes throughout the body, it will become deoxygenated. Turtles have that almost septum thing. They're very similar to the lizard, right? And that they start to make that septum to limit how much gets mixed, but you can still see that it is somewhat mixed here. But with these amphibians with the 3-chambered heart, it is mixed. Okay, because it pumps it so well, and it pumps it in a certain way that it keeps that oxygenated blood to one side of the ventricle and pumps it out. It can keep it slightly more oxygenated, but it is mixed and it is deoxygenated at that point. So not very efficient, very efficient. Okay. I will link this article into the thing so you can read it, but it kind of just talks about that. Um, there's that picture resolution, but basically talks about how it is getting mixed there. Okay. All right. I hope that answers your questions for this. This is a fairly simple, pretty quick project, and I hope that the video really clarifies and helps you answer these questions. Points. Later. Bye.